This is a GCSE biology presentation on enzymes. So to get started, have a look at the picture on the right hand side of the screen. Gives you all the main organs involved in the digestive system. And all you have to do is cast your mind back to previous presentation and explain how all those organs work together in the digestive system to digest and absorb nutrients from food. Pause the presentation now. You can do it in your head, you can write stuff down, and then whenever you're ready, you can continue on with the presentation.
The digestive system can basically be separated into two sections, the digesting part and the absorbing part. What you're looking at now is a diagram that shows all the different organs, and these ones are mostly involved with digestion. There is a little section at the start of the small intestine, which is also part of digestion, which we'll talk about in the next section. But we have mechanical digestion inside the mouth with the teeth grinding up the food. We also have salivary glands producing a substance called amylase, which is an enzyme. We'll come back to the enzymes in a different presentation. But the enzymes are what speed up chemical reactions inside living organisms and as biological catalysts. And amylase breaks down a particular substance inside the mouth. We then have the long tube that connects the mouth to the stomach, which is called the esophagus. Be careful of the spelling. The American version has an E in it, the UK version has an O at the start. We then make it to the stomach. The stomach is a large container which has hydrochloric acid along with a number of other enzymes in there as well. But the hydrochloric acid is what dissolves down the large food particles into smaller food particles. Around the stomach, we have what's called the pancreas, and the pancreas is responsible for producing a number of enzymes, for example, insulin. We also have the liver, which is responsible for the production of bile, and we have the gallbladder, which is where bile is stored in the body until it's needed. The second stage is the absorption stage. At this point, we kind of have a little overlap because the very start of the small intestines is a particular section called the duodenum. And the duodenum is the little starting section where the stomach acid that is dissolved while the food is released and mixed with a number of substances, including the bile from the gallbladder, as well as the digestive enzymes from the pancreas. And all those digestive enzymes do more digestion, and as we move through the small intestine into the ileum, we have digested food being absorbed. So the start of the small intestine is actually part of the digestion process, and it's called the duodenum. In the ileum, we have absorption of food particles, and we'll go through in more detail what exactly we mean by food particles and the substances we're talking about. We also have the large intestine, which is also known as the colon. That's where water is absorbed. We have the large intestine. We have at the end a place called the rectum, which is where feces is stored. And then we eventually lead to the anus, which is where the feces basically leaves what's called the alimentary canal. And the alimentary canal is the entire tube from the mouth all the way to the anus inside the digestive system. You need to be able to name all the different parts, and you also need to be able to identify their functions. Quite often in exams, you're given diagrams similar to the one on the right of the screen, and you're asked, what is the name of this, what is the name of this, what is the name of this? So you need to be able to identify them, and you also need to be able to name their function.
In this presentation, we're going to look at the structure of an enzyme. We're going to look at how enzymes are affected by temperature and pH, some of the properties of enzyme, and then we're going to look at calculation of rate in terms of enzyme reactions. So what are enzymes? Well, they're proteins that are known as biological catalysts. The reason they're called biological catalysts is because a catalyst is any substance that speeds up a chemical reaction, and they're called biological catalysts because they speed up chemical reactions inside living organisms, hence the word biological. They work on things called substrates. What they do is they basically get a substrate, and the substrate fits into what's called an active site. And when it fits into the active site, the chemical that is the substrate is then converted into products. You can have some enzymes, most enzymes are involved in the breakdown of substrate, but there are some enzymes that involve joining substrates together. For digestion, we're just going to focus on breaking substrates up into smaller components. So lock and key theory is simply the fact that the active site of the enzyme and the substrate that goes into the enzyme are complementary shapes. They're not the same shape, they are complementary in that they fit together perfectly. We also have a concept that enzymes are specific. Because of the complementary nature of the shapes of the active site and the enzyme, it means that each enzyme only works on one chemical reaction or one substrate. So we talk about enzymes being complementary and specific, and this is what we mean by the lock and key theory. Enzymes are affected by a number of different factors. The two main ones that we're going to look at are how enzymes are affected by temperature and how enzymes are affected by pH. When we look at temperatures at three different sections. If you look at the graph, you can see part A, B, and C. Part A is between 0 and 40 degrees Celsius, and in part A, what you'll find is that the lower the temperature, the slower the rate of reaction. And as you increase the temperature, you will increase the rate of reaction. The reason why this happens, the explanation, is because as we add more heat to the reaction, the molecules inside are moving faster and therefore we have more successful collisions. At low temperature, we have the molecules moving much slower, so we have a fewer number of successful collisions. That continues up until we reach what is called the optimum temperature. For most enzymes, the optimum temperature is between 37 and 40 degrees, and this is because 37 degrees is human body temperature. There are other enzymes that do exist at higher and lower temperatures as their optimum, but for the most part you'll be dealing with GCSE with things in around 37 to 40 degrees Celsius. Remember, the word optimum is the best one to use, but if you're stuck in an exam when you said best temperature, you can get away with that, but you should be using the word optimum. Section C, what has happened is that the temperature has been increased beyond the optimum temperature. At this point, the bonds that are holding the enzymes together are being broken, and those bonds, because they're no longer holding the enzyme structure together, result that the enzyme's active site changes shape. If the active site has changed shape, the substrate is no longer complementary to the active site, and we say that the enzyme has become denatured. So when you talk about denatured, you are talking about a change in the shape of the enzyme active site structure as a result of a factor, for example temperature, and it means that the enzyme and substrate are no longer complementary, and if they're no longer complementary, they no longer stick together and form what is called an enzyme substrate complex. And that's the name given when the enzyme and the substrate are held together in place. Enzyme substrate complex, and if we've denatured the enzyme, they no longer exist. As I said, the second thing that we need to look at in terms of enzymes is the effect of pH on enzymes. And you can see the graph is a different shape. This is because we have an optimum pH. Remember, optimum means the pH at which the enzyme will have the fastest or highest rate of reaction. So at the optimum, we have the highest rate of reaction. If we increase the pH above that optimum or below that optimum, then we have a denaturing of the enzyme. Remember, when the enzyme denatures, the enzyme has bonds broken. It no longer holds its shape. The active site has changed shape. The substrate is no longer complementary, and an enzyme substrate complex cannot form. And you have to get used to those sentences, folks, because that's what it's going to ask you for in a big 5-6 marker exam. You'll have to explain the connection between pH and temperature, the fact that it's above or below optimum in pH, or if it's temperature, it's above the optimum. We talk about denaturing, we talk about the breaking of bonds, we talk about the change of structure, the active site is no longer complementary, and enzyme substrate complexes cannot form. One of the questions you might get around this topic is a calculation question asking you to work out the rate of a reaction, and then quite often they'll ask you to put that into a graph. Now, in terms of rate of reaction, there's two main ways that you can calculate the rate of a reaction in enzyme reactions. The first way is you calculate how quickly substrate is used or how quickly a product is formed. And you do that by getting the amount of reactant used and dividing that by the time, or you get that by the amount of product formed and divide that by the time. Note the use of the word amount. In your GCSEs, you should not be using the word amount. It should always be volume or mass or length or distance. It always should be a specific value. We can use amount here because depending on the question, they may say volume or they may say mass. But you need to be very careful when you're doing your calculations that you should use exactly what's on either the results table or the graph that they've given you to use in the question. So you get the amount of reactant, the mass of the volume, and divide that by the time, or the amount of product formed, again, either its mass or its volume, and you divide that by time to get the rate of reaction. Now the second way you can do it is if you have a chemical reaction, an enzyme reaction, where there's a clear endpoint. So for example, you put A and B together, and when the reaction ends, it changes from pink to brown, or some other color change or some other variation thereof. If you have a clear endpoint that you can see when the reaction has happened, you can time how long it takes for a reaction to occur. You can use the equation 1 divided by time for a reaction to occur as an indication of the rate of a reaction. So those are the two ways you can calculate the rate should they ask you that question in an exam. Just to give you an example, in this question it says an enzyme chemical reaction, A and B react to form C and D, calculate the rate of a reaction in the first 30 seconds. Well, it doesn't really matter about the reaction A, B, C, and D. We just need to know that D is a product, and it tells us that in the first 30 seconds from the graph, again, for good practice, draw lines on the graph in your exam, because if you make a mistake, you might still steal a mark. But it's easier to read if you've drawn lines with a ruler and a pencil. If you haven't drawn the lines, it's very easy to misread. So drawing lines makes you more likely to get the correct answer. And it says in the first 30 seconds, we go across and we have 10 cm3 of gas produced. So we simply get, in this case, the volume of D produced, divide that by the time, which is 30 seconds, and that gives us a value of 0.333 repeating. It does say give your answer to 1 decimal place, so I've rounded it off to 0.3. Last thing to finish off is you have to go and talk about everything that's been covered in this presentation for a grand total of 2 minutes without stopping. If there's any repetition, if there's a pause, if there's a mistake, anything goes wrong, you have to start your 2 minutes again. Now you can do this by talking to a family friend, from a member, you can do this by talking to your pet, you can do it by talking into the mirror, or just simply talking to yourself. It's not a problem. You've got to be able to talk about it for 2 minutes. In order to be able to talk about it for 2 minutes, you have to go back and make sure you've memorized some of the content.